Hey y'all, welcome to Kelvin's Classroom. Thanks for coming back. Today we're going to be talking about food tests. Now, food tests are things that we can do to look at substances and see what's in it. Now, we can do lots of tests on a lot of different substances, and there's lots of different ways we can test these. We're going to just look at some of the really simple ones that you need to know for GCSE level to understand our 4 main food groups that we look at. Now, last time we talked, I said there were 3 main food groups. One of them we're going to split into 2, and we're going to kind of talk about why that is. So first, we're going to talk about what those groups are, what the tests are for them, and what a positive result would look like in each of those. And then the second thing we're going to do is we're going to look at why this matters, when we might do this, when we might do some of these tests in a more real life type situation. And then the third and final thing we're going to talk about is another way of testing food, not to find out what substance is in it, but another type of food test that we could do. All right, so let's go ahead and dive on in. So the food tests that we do are the Benedict solution test, Biuret solution, iodine, and then our last one, which is either ethanol or Sudan III, are the two substances that we most commonly use for it. Now, these are ones you're just gonna have to memorize what they are, and I'm gonna help you guys figure that out as we move along. So first thing, let's figure out which test tests for which substance. So we need to know what substances we're going to be testing for. So we talked about having carbohydrates, proteins, and lipids as our 3, right? So we're going to do a protein test. We're going to do a lipid test. With carbohydrates, we're gonna look at 2 different types of carbohydrates. We're gonna look at starch, and we're gonna look at simple sugars. So once we've broken those starch down. So 4 tests we'll do: starch, sugar, protein, and lipids. Now, how can we remember and match up the name of the test to the actual thing that we're testing for? Well, let's start by looking at iodine. Iodine, usually a brownish, orangish type substance. Now, I can't give you a great hint as to how you can remember which one this tests for, but this is a really simple one to test for because it's testing for starch. And if we think about a nice white piece of bread, right, if we drop that iodine onto it, it's going to turn a very dark black or dark purple color. And that tells me, yes, starch is present. Right? Now, as we see, very simple test. All we have to do is just add it directly to the substance and it can tell us this. Or if we're testing it for something inside a test tube, we just mix the solution up, add a couple drops of iodine to it. Swirl it gently, look at it, and a positive result will be black or dark purple, and a negative result will be orange brownish color. All right, excellent. So that's iodine test. That's one out of the way. That is probably one of the simplest ones that we have. Let's look at our next one, which is our glucose test, right? So we tested for starch. Now let's test for what it looks like once we've broken it down. Now, if we're testing for glucose, a starch test will not test positive for it, right? So if I have simple sugar and I add the iodine to it, it's not going to test positive. It's going to be negative. But if I do my sugar test, it'll be positive. What our sugar test is Benedict solution. Now, this is where we're going to have to come up with a solution of how we're going to try and remember this. Benedict solution. The way I like to remember it is I'm a Marvel fan. I like to think of Benedict Cumberbatch. Benedict Cumberbatch, who I think of for this one. And now I think about him since he's Doctor Strange. I imagine doing magic all the time like that probably takes a lot of energy. It takes a lot of energy, and he probably gets really tired doing that. So I imagine he probably needs a quick boost of energy regularly. And sugar is how we get that boost of energy. So we can think of Benedict Cumberbatch, sugar, they go together very well. That's the test itself. I had someone once tell me that they actually prefer to think of Benedict Cumberbatch as being a really sweet guy. So they think about it being sugar, Benedict Cumberbatch, Benedict solution, sugar. They go together. Anyway, using the Doctor Strange example, when I imagine him doing that magic, what color is it? Usually we see those orange, red, maybe yellow flames coming out as he's doing magic. This is going to help us looking at the test, because when I'm doing a Benedict solution test, a positive result is going to be red, orange, yellow, one of those colors. They tell me that it's there. Now, Benedict solution starts with B, Benedict, and our solution is blue to start. So when we're doing a test, we take our Benedict solution, which looks blue, we add it to a test tube mixed with the solutions we're trying to test. Right? We gently swirl them together. Now, if I look initially, I don't see any change. None of the solutions that I have are going to change. That's because this takes one extra step to our other test. We need to add heat. If you're not sure how you're going to remember that, let's go back to that Doctor Strange thing. We're going to keep going with it until it's run out of any juice. If I look at the magic portals that he's opening, I look at those sparks, I think they probably would give off some heat, right? Sparks give off heat. I add heat to my Benedict solution. I have to add heat to it. Now, once I've added heat to my Benedict solution, I pull it out. I can look at them. If it's still blue, then it's negative. There's no sugar present. If it's green, now that's telling me that there is sugar present, but it's just a tiny bit of it. And then if it moves into yellow, there's more, and orange, there's more, and dark red is saying there's a lot of it present. So this is one of those tests that tells us whether something is present or not, but it also kind of helps us know how much of it's present, okay? We can use this to see if there's a lot of sugar, a little sugar, or no sugar present. Cool. So we've got two down. We've got starch, we've got sugar. Let's move into our next one. Let's move into protein. When I think of protein, I think back to Texas. I think beef. That's what I grew up as eating, beef as my type of protein. Biuret, beef, they kind of go together. They seem similar. They both start with a B, I guess. I don't know. That's the only way you can try and remember that one. If you've got a better idea, throw it down in the chat. I'd love to have you guys hints and helps with this one as well. Now, just like Benedict solution starts with a B and is blue, Biuret solution starts with a B and it's blue as well, right? Now for this one, we take our Biuret solution, we add it to the test tube where we already have our solution mixed up. If it stays blue, after we swirl it just a little bit, if it stays blue, then it's negative. If it turns a purple color, then that means that there's protein present. So purple, protein, some people might refer to it as a lilac color, right? But we're looking for that change of color, and that means that protein is present. Brilliant. We're on to our last one that we're going to be testing today. There are other ones that we're going to learn in the future, but for now we're going to stick to just these 4 food tests, and that is our test for lipids. Now, with this one, ethanol is probably the easier of the 2 solutions that you use, but it works the same with Sudan. I'm going to show you a demonstration using both of them so you can kind of see what it looks like. Now, for this one, the way that I remember this one is lipids. A lot of people think of it as fats. Ethanol is a type of alcohol. So if I were to drink too much alcohol, I would start to gain extra fat. Alcohol, fat, ethanol, ethanol and lipids. They go together. Now, ethanol starts as a clear solution. I add it to my solution here. I need to add a little bit of water as well to this one so that way I can create a barrier, create a little bit of separation. When I swirl them together, now if there's nothing present, I'm not going to see anything happen. It's just going to stay as it is. It's going to be a clear solution. But if there is lipids present, I'll start to see this little cloudy layer right here at the top form that precipitate. That's telling me that there is a lipid present in that substance. With Sudan III is a red solution. I go through all the same steps, nothing different. The only difference is now for a positive result, I'm looking for this red mixture, cloudy bit up here at the top. There we go. That's the precipitate I'm looking for if it's a positive result with Sudan III. Right. So those are our food tests. Pretty simple, not too complicated, not too hard. Try and remember which one tests for which. You have to practice those until you can just get them logged into your memory and remember them. I did say there was one other test that I was going to mention. Now this is a calorimetry test. Calorimetry, like a calorie, not like color. Don't mix those up because there is colorimetry, which is a different type of test. This one, calorimetry. Now this is how we measure how many calories or how much energy is stored inside food. For this one, all we do is we burn a food. We measure how much heat is given off by it, how much energy is given off by it. We compare it to how much mass is lost, and we can use that to calculate how many calories are in different foods, so how much density is in each food. All right, now, when are we ever going to use any of these tests in real life? Well, the calorimetry one is good for a lot of food scientists will use it to measure different food density, things like that. But the other tests can be used for a lot of things. Now, the main one we're probably going to see used is testing for glucose. Now, we can test different things for glucose and how much glucose they have to monitor things like blood sugar levels, right? That's really important. We can also use the protein one if we want to test if there's protein inside urine or anything like that. We can see a lot of health concerns surrounding things like that. Inside more of a practical application but science setting for you guys, one time that we normally see it is we will take tubing that is very similar to small intestines, and we can mix an enzyme and a starch substance on the inside of it. Now, on the outside of it, there won't be any starch. There will just be water. There won't be any sugar either. We can test and prove that by looking at this. We can test it there, see that there is nothing, there's no sugar present, no starch present. Now, after time passes, we can take another sample of it and we can see that actually some sugar's present. What this means is the starch inside the tubing was broken down by the enzyme into sugar, and the sugar was able to dissolve across there. It was able to move and diffuse across that tubing, just like it does inside the small intestines. This is a way we can test different digestion, look at how enzymes can affect the rate of reaction, how different temperatures can affect the rate of reaction. This is one of the ways that we can use these in a more practical sense. All right, thank you guys so much for coming. Today's was a quick one looking at food tests. It's important to be able to understand these, to be able to test these. I'm sure you guys are going to test these out in your classroom, so have a go at it. Hopefully you understand it before you get there. Come back next week. We're going to move on to a new system. We're going to start talking about the circulatory system. So come on back and we'll do that. Thanks.